Let's take a look at the nucleosides, nucleotides, and nucleic acids, 核苷酸 and 核酸. So first, let's take a look at nucleosides. Molecules containing a sugar moiety and a non-containing base moiety. This is the nucleosides. 核苷还有糖部分和含氮碱基部分的分子。In RNA, a monosaccharide is idopentose called D-ribose. In DNA, the monosaccharide is D2 deoxyribose. The nandopentose that lacks a hydroxy group at C2 position. Take a look at the structure. You can see the difference. This is the ribose. This is the C2 position is hydroxy group here. On this side is the 2-deoxyribose. At the C2 position, you cannot see a hydroxy group here. The hydroxy group is missing. Is why it's called a deoxy. That's the why the name of this deoxyribose. 在 RNA 中，单糖是戊醛糖低核糖；在 DNA 中，单糖是D2脱氧核糖，一种在 C2 的位置上没有羟基的戊醛糖。Only 5 common nitrogen-containing bases are present in nucleic acids. 3 bases with 1 ring, cytosine, uracil, and thymine, are derived from the parent compound pyrimidine. 2 bases with 2 rings, adenine and guanine, are derived from the parent compound purine. Each base is designated by 1 letter abbreviation. 核酸中有5个常见的含氮碱基，3个带1个环的碱基，泡嘧啶、尿嘧啶和雄腺嘧啶，衍生自母体化合物嘧啶。具有2个环的碱基腺嘌呤、尿嘌呤衍生自母体化合物嘌呤，每个碱基由一个缩写英文字母来代表。Take a look at the structure. This is a pyrimidine, 6-member ring containing 2 nitrogens and 4 carbons. This is cytosine. Cytosine has 2 substituents. One is the amino group here, another is the carbonyl group. Uracil has 2 carbonyl groups on this pyrimidine ring. Thymine has a methyl group here and 2 carbonyl groups on the pyrimidine ring. This is the purine. Purine is a 2-ring compound with 4 nitrogen in the rings. This is the adenine. Adenine has an amino group on the purine compound. Guanine has a ammonium group here and a carbonyl group here. A nucleoside is formed by joining an anomeric carbon of the monosaccharide with the nitrogen atom of the base. A nucleoside is a non-glycoside because it's a nitrogen derivative of the glycosides. Primes, the symbol like this, are used to number the carbons of monosaccharides in the nucleosides. in a nucleoside to distinguish them from the carbon atoms in the rings of the base. 通过将单糖的一头碳原子与碱基的氮原子连接形成核苷。核苷是一种单糖苷，因为它是糖苷的单衍生物。撇号用于对核苷中的单糖的碳进行编号，以将它们与碱基环中的碳原子区分开来。With the pyrimidine bases, the nitrogen atom at the 1 position bonds with the 1' carbon of the sugar. With the purine bases, the nitrogen atom at the 9 position bonds with the 1' carbon of the sugar. 对于嘧啶碱基1位的氮原子与糖的1撇碳键合。对于嘌呤碱基9位上的氮原子与糖的1撇键合。Take a look at the structures here. This one is D-ribose. This is C2 position. You can see the hydroxy group here. This is C1 position. It's also called an anomeric carbon. Here, this one. This carbon forms a bond with the N1 on the base. This pyrimidine, this is cytosine. This N1 here, and after the form the bond, this compound is called cytidine. And you can see this new bond here. Next, you can see this D2 deoxyribose. You don't see a hydroxy group at the C2 position. This anomeric carbon, also they called C1 carbon of the sugar. This is the N9 position, nitrogen 9. So they form the new bond between this C1 on the sugar and N9 on the base. This is the adenine and this base. Then this new compound is called deoxyribonucleoside. It looks like this. Next, let's take a look at how to name nucleoside. 命名核苷。To name a nucleoside derived from a pyrimidine base, use suffix -ide, -ine. The example is here. Cytosine becomes cytidine. 在命名衍生自嘧啶碱基的核苷时，使用后缀 -ide, -ine. To name a nucleoside derived from the purine base, using suffix -o-s-i-n-e. Example is here. Adenine becomes adenosine. 在命名源自嘌呤碱基的核苷时，使用后缀 -o-s-i-n-e. For deoxyribonucleosides, add the prefix deoxy, as in deoxyadenosine. 对于脱氧核糖核苷添加前缀 deoxy， 比如 deoxyadenosine. Next, let's take a look at the nucleotides. Joining nucleoside with a phosphate group. 核苷酸将核苷与磷酸基团连接。Nucleotides are formed by adding a phosphate group to the 5' hydroxy group of the nuclear side. Ribonucleotides are derived from ribose, while deoxyribose nucleotides are derived from 2-deoxyribose. Nucleotides are named by adding the term 5' monophosphate to the name of the nucleoside from which they are derived. Abbreviations are used for nucleotides. For example, cytidine 5' monophosphate is abbreviated as CMP. It's a CMP. And deoxyribonucleoside 5' monophosphate is DAMP. You can say DAMP。核苷酸是通过在核苷的无皮的羟基上添加一个磷酸集团而形成的。核糖核苷酸含有核糖，而脱氧核糖核苷酸含有2脱氧核糖。核苷酸是通过其衍生的核苷名称后，在后边加上无皮monophosphate来命名的。核苷酸通常用缩写字母，比如说单磷酸胞苷。单磷酸胞苷 is a cytidine 5' monophosphate 缩写成CMP。单磷酸脱糖腺苷Deoxyribonucleoside 5' monophosphate 缩写成为 DAMP。2 or 3 phosphate groups are added to nucleosides. To name those compounds, add the suffix diphosphate or triphosphate to corresponding nucleoside name. For example, adenosine 5' diphosphate ADP and adenosine 5' triphosphate ATP。2个或3个磷酸集团被添加在核苷上，在命名这些化合物时，在相应的核苷名称中加上后缀 diphosphate 或者说triphosphate。The example is adenosine 5' 无皮 diphosphate ADP is ADP, and adenosine 5' triphosphate ATP. This is the structure of ATP. This is 3 phosphate group here. This is derivatives. This adenine base here. Next, let's take a look at those structures here. This one. This phosphate. This is a cytidine. Ribonucleoside. This is a ribonucleoside. It's a phosphate. Then they link the phosphate to the C5. This 1, 2, 3, 4 C5 position. C5 prime. This is sugar here. And then they form this new compound. They call the cytidine 5' monophosphate. Monophosphate means that it's only one phosphate here. This is another example. This is a deoxyribonucleoside. Here is you can see this base. This is the sugar at C2 position. The no hydroxy group is called deoxy. This deoxyribonucleoside. This whole thing. It's called deoxide. It's a phosphate, and the new products, and the same thing. The phosphate is bonded to the C5' position here, the new compound is called deoxyadenosine 5' monophosphate. Only one phosphate. Okay, next, let's take a look at the nucleic acids, DNA and RNA. Nucleic acids are single chains of polymers of repeating monomers called nucleotides. In other words, nucleotides are monomers of nucleic acid. 核酸是以核苷酸为单体的聚合链。There are two types nucleic acid. 两种核酸，有两种核酸。The first one is the DNA. The second one is the RNA. DNA, Deoxyribonucleic acid, store genetic information of organism. DNA 的脱氧核糖核酸储存着生物体的遗传信息。RNA, ribonucleic acid, with a major function of transmitting genetic information for protein synthesis, among many other functions. RNA 核糖核酸具有传递蛋白质合成遗传信息的主要功能，此外还有许多其他功能。Nucleic acids are formed by joining the 3' hydroxy group of one nucleotide with the 5' phosphate group of the second nucleotide in a diphosphodiester linkage. 核酸是通过将一个核苷酸的3' 的羟基集团与第二个核苷酸5' 的磷酸集团以2磷酸2支键连接而形成。Take a look at how this nucleic acid formed, and the first nucleotide is a cytidine. Cytidine is a cytosine, is a phosphate. This is the sugar. D-ribose is a phosphate, and this is a 5' phosphate. There is a 3' hydroxy group form a bond with this 5' phosphate of the second nucleotide. This is the new bond here. And then this one is the second nucleotide form a bond with the third nucleotide here, and here, and so on. Then this whole chain is called nucleic acid. The last part, let's take a look at the characteristics of nucleic acids. 核酸的特性。A polynucleotide contains a backbone consisting of alternating sugar and phosphate groups. 多核苷酸主链是由交替的糖和磷酸集团而组成的。Identity and order of the base distinguish one nucleotide from another. 碱基的特性和顺序将一种核苷酸与另一种核苷酸区别开来。A polynucleotide has one free phosphate group at the 5' end and a free hydroxy group at the 3' end. 多核苷酸在5' 端有一个游离的磷酸集团，在3' 端有一个游离的羟基。 One of the differences between DNA and RNA is that the sugar molecules in DNA is the deoxyribose, which lacks the hydroxy group at the C2 position, while the sugar molecules in RNA is the D-ribose. DNA 和 RNA 的区别之一是 DNA 中的糖分子是D2 脱氧核糖。D2 脱氧核糖在 C2 的位置上没有羟基，而 RNA 当中的糖分子是 D 核糖。Another difference between DNA and RNA is that DNA contains thymine and RNA contains uracil, in addition to 3 common bases, adenine, guanine, and cytosine. DNA and RNA 之间的另一个区别是呢，除了腺嘌呤、adenine、鸟嘌呤、guanine和泡嘧啶、cytosine 这3个共同的碱基之外呢， DNA 含有特定的雄腺嘧啶 thymine， 而 RNA 含有特定的尿嘧啶 uracil。 In summary, DNA contains bases A, G, C, and T. DNA 含有碱基 A, G, C, T. RNA contains bases A, G, C, U. RNA 包含碱基 A, G, C, U。